guys, welcome to chapter 6. I just finished up the remainder part of 5 and wanted to remind you that there's only going to be probably one, maybe two questions from the remainder of that chapter. So this is really technically not the beginning of exam 2, but for the sake of being a full chapter, this is the beginning of the material for exam 2. It's going to be over chapter 6, 7, 8. We do not do chapter 9, and then finish up with 10 for exam 2. All right, let me move myself out of the way there. Chapter 6 is all about microbial growth, and the great thing about microbial growth, depending on whether or not you're looking at that from a human aspect or if you're looking at that from a microbial aspect, one of them is a good thing and one of them is the bad thing. We are going to look at this from a microbial aspect since this is microbiology, and so microbial growth is something that's essential for a microbe to be able to survive. If microbes grow, we're not talking about microbes getting fat or getting taller. That's not how it happens. I'm talking about them increasing in the number of cells that they have. In order to be able to divide, microbes, of course, do need a few things, just like you do. You need oxygen, and you need a stable internal temperature, and you need water. There's lots of things that you need, necessity-wise, in order to be able to survive. Microbes are no different. Microbes need physical and chemical properties that are able to allow them to be able to grow and therefore survive. We're going to look at the physical requirements first. Most of the time, microbes really don't like it too cold or too hot. That's the thing. Mesophiles are right there in the middle. I remember mesophiles love me. Moderate temperatures, right there at your body temperature, that's what mesophiles like. And a very large group of microbes fit into that category of mesophiles. There are some, though, that are on the outer ends. So some of them actually like the extreme temperatures where it's actually rather cold. Those are psychrophiles. Psychrophiles live in places like your refrigerator. People, your refrigerator does not kill bacteria. I see so many people who do not store food properly. I go over to people's houses and, I don't go through the refrigerator, but I mean, they'll open it up and I just kind of am standing there and I see things open and just kind of laying in the refrigerator. We have plenty of microbes that survive just fine in colder types of temperatures. We used to think that microbes couldn't survive down in the depths of the ocean because it was too cold for them, and now we know that that essentially is not true. There are psychrophiles that actually love that temperature. And then there are thermophiles. If I was a microbe, I would be a thermophile because I feel like I should not have to live in temperatures below 80 degrees. But thermophiles love the heat. They love to have those warmer types of temperatures, and those are what they seek out and thrive at when they're surviving. When you look at other requirements, microbes need a steady pH. Most microbes grow in between 6.5 and 7.5, somewhere in there. They don't like it too acidic. They don't like it too basic. On the other hand, there are molds and yeasts that will grow right in between 5 and 6. There are some microbes that actually do like acidic types of environments, and one of the ones that is notorious for that is called Lactobacillus. Lactobacillus is an acetophile, meaning it loves acid. And Lactobacillus is cultured naturally. That means it's supposed to be there. It's the normal microbiota inside of the vaginal cavity. The vagina is a very difficult place to colonize. We talked about this because it's warm, it's moist, it's acidic. Lactobacillus actually have a job, though, inside of the vagina. And you can see this whenever a female takes an antibiotic and wipes out not only the bad bacteria, but also the good bacteria. Whenever you wipe out those good bacteria, the vaginal cavity is somewhere acidity-wise at 5.5. So if I take out the lactobacillus, 5.5 is right in the prime growth for what? Yeast. And that's what happens. Lots of women who take antibiotics have a yeast infection because they rid their body of the lactobacillus. And when lactobacillus is gone, yeast has a field day. And so we a lot of times give what are called acidophilus pills. Those are probiotics that a lot of people take in order to rebalance that pH. Those same type of acetophiles are found in yogurts. They're found in sauerkrauts and pickles. So a lot of times women will eat yogurt, especially something like Activia, where it has a high content of the probiotics. And they'll eat that in order to replenish some of those natural types of microbes. We also need to make sure that the environment surrounding the microbe has enough pressure and that it's not too much salt or too much sugar. A little bit of salt goes a long way, right? Have you ever been cooking and maybe you're not paying attention, or maybe you don't know exactly how much salt to put in? You kind of shake a little bit of salt in there and you get too much and you know it. And it doesn't take a lot to overdo it. So a little bit of salt goes a very, very long way. And in microbes, most of the time, if you place a cell inside of that hypertonic type of solution, the cell will dehydrate and it will shrivel up and it will plasmolyze, which means that the plasma membrane gets pulled away. And essentially that cell would die. But there are some microbes who actually do love the salty types of environments, and they live quite nicely within it. So it seems like when you look at the microbes, there's always that middle area where people or microbes want to be. And then there's those extreme types of cases that can be sort of exception to the rules. When you look at chemical types of requirements, microbes have to have a carbon source. They have to have energy. They've got to utilize that carbon source in some way for energy production. They need nitrogen. They need sulfur. They need phosphorus. They need those trace types of elements like zinc and copper and iron. But let me say something about those trace elements. We actually don't have to worry about those, because when we're mixing up media for the microbes to grow on, tap water has enough of these trace elements that we don't have to worry about giving them, since microbes are so small and they need such a small type of content. It's interesting when you look at microbes and you view how they grow in a test tube. One of the things that I used to do in a microbiology lab is I would set up 6 or 7 test tubes, and my job essentially was to incubate these bacteria and then look at whether or not they were aerobic or anaerobic or facultatively anaerobic, because we needed to know whether or not they consumed oxygen. When we look at this slide that you're looking at here on the screen, it gives the different examples of a test tube and what you would be looking for if microbes were able to utilize oxygen or not use oxygen. And so let's take a look at these, because this is something that I will want you to know. In that first one labeled A, one of the things that you notice is that all of the microbes are growing in a clump right at the top of the test tube. Not in the middle, not at the bottom, only at the top. The reason why they're growing there is because that is where the highest amount of oxygen is found within that test tube. So those are obligate aerobes. Obligate aerobes have to have oxygen. There's no way around it. Next up is B. Now, one of the things that you see in B is up at the top of that test tube, you do see a ring of growth. And then you see a disbursement of microbes growing sort of throughout that test tube. Well, when you look at this test tube, the first thing that I see is in fact that ring around the top. And that tells me that those microbes used the oxygen present. But the fact that there is microbes present throughout tells me that they didn't stop growing when the oxygen was gone. So facultative anaerobes absolutely use that oxygen up at the top first, but then when it's gone, they will continue to grow throughout the test tube. On C, we see growth down at the very, very bottom and nowhere else. That's because those are obligate anaerobes. Obligate anaerobes have to grow in the absence of oxygen. If I need to take away all of the oxygen, I need to drop way down to the bottom, and that's the only place that those are going to grow. D is an aerotolerant anaerobe, and what I notice about D that is different from B is there is no growth ring around the top. What I see are microbes dispersed all throughout that test tube, and it's pretty even. I've got some down at the bottom, I've got some in the middle, I've got some at the top. That's because aerotolerant anaerobes pretty much ignore oxygen. They could care less if it's there or not. If it's there, it's there. If it's not, it's not. So they tolerate the fact that oxygen is there, but they're not going to utilize that oxygen up at the top like a facultative anaerobe. And lastly is a microaerophile. Microaerophiles grow right in the middle. And the reason why is because they do need oxygen, but what's the word micro mean? Little or small. So they need a smaller amount of oxygen in order to grow. So what we find is that that band right across the center is going to be these microaerophiles, because up at the top, that's too much oxygen. Down at the bottom, that's too little oxygen. They need about right in the center of that. And again, we would do this with an unknown type of bacteria, because knowing whether or not it needs oxygen and the amount of oxygen it needs can help me sometimes to identify organisms. Sometimes, though, oxygen can actually be boosted into a higher state, and at that time we can actually form what are called free radicals. The most reactive type of free radical has got to be the hydroxyl radical. The hydroxyl radical, that OH radical, is very, very reactive. On this slide that you're looking at, I think you're looking at it. I can't see the slides that you're looking at, but yes, you are looking at it. Okay, so on the slide that you are looking at, I want to point out two things. You see right there in the center where it says catalase and peroxidase. The peroxide anion is interesting because H2O2, which is hydrogen peroxide, is something that a lot of people have a misconception about. I remember when I was younger, my mother I would fall, scrape my knee, whatever, and my mother would set me up on the sink, and she would take out her hydrogen peroxide, and she would pour it on my knee, and she would say, "Look, you can see that it's working." Why did she say that? Why did she say you could see it killing all the nasty bacteria? Because it bubbles, right? Have you ever done that? You put hydrogen peroxide on a cut and it kind of bubbles up a little bit? Okay, well, let me tell you the truth about it. Many microbes actually have enzymes like catalase and peroxidase that take hydrogen peroxide and convert that into water and oxygen. Let's look at catalase. If a microbe has catalase and you place it in hydrogen peroxide, it breaks that down into water and oxygen gas. If I go underwater, holding my breath, down to the bottom of a pool, and you're standing up at the top of the pool, and I blow out all of the air in my lungs, what do you see at the top of the pool surface? You see bubbles, right? Because whenever you release oxygen inside of water, it bubbles. So actually, when you put hydrogen peroxide on a wound and it's bubbling, that's the microbes laughing at you. The microbes have taken hydrogen peroxide, they have converted that down into water and oxygen, and that's why it bubbles. So we don't even really use hydrogen peroxide anymore in medicine. If you have worked in a doctor's office or in a clinic or in a hospital, it's really not found a lot. It's wonderful for irrigating wounds. Like if you fall and there's gravel, a lot of kids have, bike wrecks or something, and maybe there's some gravel or dirt in there. It's fantastic to irrigate that wound out and flush all that out. But as far as being antimicrobial, not so much. So if your mom or grandma is talking that it's bubbling, no, that doesn't mean it's working. We also see that in order to survive, microbes do need vitamins, they do need amino acids in order to make their proteins, and they do need things that they must get from the environment. If we're going to grow microbes, not if, when we grow microbes in lab, we have to provide them some sort of medium in order to grow upon. Because a medium is essentially a palette that is going to give the microbe all the nutrients that they need. It's already prepared, it's right there, ready for them to grow. If I say that I'm looking at a culture, what that means is I'm looking at a specific medium that has already grown microbes. Something that is sterile would have no living microbes on it. Now that doesn't mean that the microbes, you can't see anything. It doesn't mean it's clear. Sterile a lot of times means that I've either maybe put it in the autoclave, or maybe I've boiled it to sterilize it. So the microbes were there. We've just killed them out. We are going to use in our lab nutrient agar. Nutrient agar is essentially a very complex polysaccharide that's linked together that you can pour into a petri dish. We use this for a couple of reasons. Number one, it's a buffet for microbes. They absolutely eat nutrient agar up. All of the nutritional, anything that they would require nutritional wise is found within agar. Another great thing about agar, though, is that it is a solidifying agent. So when we pour these into our plates, it's actually liquid. But let it cool down and it turns gelatinous. It solidifies. You can flip it upside down. And the great thing about that is that microbes will grow on top of the agar. So I can provide them not only with the nutrition, but with a platform for them to grow upon. That way, when I go to look at the microbes, I know exactly where to find them. They're going to be growing right on top of that agar that we have used. Agar is not the only culture media out there. There are lots of culture medias, and in our class we'll do two. You all will make nutrient agar. It comes in a powder. We have to mix it up, and we have to boil it, and then we have to pour it and let it solidify. So you guys will be making nutrient agar. But there's also the other one that we use. It comes already poured, and it's called blood agar plates. So essentially they spin down red blood cells, and they put that into an agar, and what that does is whatever we inoculate, which remember that means to put, so whatever we inoculate into the blood agar plates essentially would mimic how those microbes would grow in your bloodstream. So that's a good thing that we can do as far as swabbing throat cultures or nose cultures to see how microbes actually would grow inside of your bloodstream. Those are the only two that we use, though. But if I'm being honest with you, there's lots of different culture media, and I just want to expose you to these, very similar to what we did with the microscopes. So you won't be seeing this media per se, but it's good that you know what it is. So a chemically defined media is a media that we know exactly what goes in it. And the reason why I need to do that is because some microbes are called fastidious, which means I have to feed them certain things if I want them to grow. For example, there is a bacteria called Haemophilus influenza. Haemophilus influenza causes all kinds of things. I mean, it can cause pneumonia, it can cause a skin rash, it can cause an ear infection, it can cause meningitis, it can cause epiglottitis. There's kind of a large range of where we see Haemophilus at. But the thing about Haemophilus is if I just put it on regular agar, it does not grow. I have to give it two things. Number one is factor V, and number 2 is factor 10. Those two things are required for that particular microbe to grow. And so if I'm wanting to grow Haemophilus influenza, I have to make sure that I put those two factors inside of the agar. So that's a chemically defined media. A complex media is what nutrient agar is. It's kind of a smorgasbord. It's just like a slop fest, if you will, for microbes. Like, here you go, here's your stuff, eat it up and enjoy. So we don't really know exactly, technically, everything that may be going into it. If I want to grow anaerobic types of bacteria, I have to be very cognitive about the fact that anaerobes can't grow in oxygen. Well, that means I would have to get rid of the oxygen on the media if I'm going to grow those. Most of the time we do that by adding a chemical, and usually it's sodium thioglycolate. I mean, I could add something like cyanide, but we technically don't play with cyanide. So sodium thioglycolate is what we usually use, and we put that in the media, and what it does is it combines to all of the oxygen gas and depletes it out. We also have to make sure that we're very careful about the cultures when we're growing them anaerobically, because we have to make sure no air can leak into it. We have to make sure we heat it to drive off any extra oxygen. We can't expose it to the air at any time. So growing anaerobic bacteria sometimes is a little difficult. We do have different ways that we can grow them. Here you see an anaerobic jar. Here you see, this is what I use. I think you're looking at a chamber. Yes, this is an anaerobic chamber, which is what I use. And you essentially have ports for your hands to go in that is airlocked, so you never actually expose the chemicals and the microbes to the environment outside. It's always in an airproof type of chamber. You can do something very simple. Most hospitals use those little packets down at the bottom called CO2 packets. You break a CO2 container and you shake it up, and that depletes all the oxygen inside of that packet, and that packet is nice and sealed off, so those petri dishes that are inside of the packet would stay and be able to grow anaerobic microbes. Sometimes we can utilize what is called selective medias. There are some times that I want to encourage certain microbes to grow, and for everything else, I want to suppress them. For example, if I were to be looking at your gut microbes for a specific microbe, there are tons of microbes inside of the intestines. There would be very, very difficult time if I was trying to isolate one particular type of bacteria when I have all of these bacteria flourishing. So one of the things that we can do, and I believe the example up here is for Salmonella, if I wanted to encourage Salmonella to grow and I wanted to inhibit everything else, I could put it on a very selective media. A bismuth sulfite agar can isolate Salmonella, and what it does is nothing else from the gut grows on that. So if I suspect that you have Salmonella, I could take a fecal swab. I could swab it on the bismuth sulfite agar, and I could incubate that. If something grows, that tells me that you have Salmonella. If nothing grows, I don't know what you have, but I do know you don't have Salmonella. So it's a way that we can help not only to identify certain organisms, but I also can suppress everything else from growing. I can do a differential media. One of the things you'll notice when you get in lab is when you're looking at the plates, everything kind of looks the same, like a little white dot, or sometimes you'll get a little yellow or a little pink dot, but it all looks the same even though they're very different microbes. That's because they don't grow differently on agar. If I put them on a differential media though, every single colony of microbes would look different. So on this slide, those big dark black circles, all of those are the same kind of microbes. And then those little tiny red colonies that you see, all of those are the same colony. So what it does is it allows me to visualize that, okay, I have one type of bacteria growing here, I have another type of bacteria growing here. We also have enrichment types of medias. enrichment types of medias are going to encourage the growth of certain microbes. Now, the thing that's different about this from selective media is we're not necessarily discouraging everything else. We're simply encouraging one specific type. So I would add things to the media to see if they would be able to grow in the presence. With microbes, we want to make sure that we can study colonies. I want to look at a pure culture because that means that those cells all have one specific strain. And if I can isolate one specific strain, if I'm trying to diagnose someone or if I'm trying to identify what a microorganism actually is, then a pure culture is what I need to work with. So whenever we're making these pure cultures, essentially what we see is that we're able to streak a plate and isolate a specific type of colony. And what that does for us is allow us to be able to look at those colonies and say, okay, well what could this potentially be? I'm going to teach you this technique that you see on the plate here of streaking, and we'll be doing this together in the lab, talking about it a little bit more. But essentially what we're doing is, if you look at the concept of it, we're diluting down this sample so that we can get individual colonies in order for us to count. Sometimes I want to make sure that we can preserve our bacteria. A lot of times in lab, I will keep them just in a refrigerator. I mean, it's no different from the refrigerator that you have at home, and they'll keep for quite a bit. I know it says short-term storage up here, but they'll keep for about a year that way. Usually they dehydrate out because the water will, dry up and they'll dehydrate. If I want to keep them for a long term, though, I can actually do something like deep freezing, or I can freeze dry them, and essentially they can last forever that way. We've talked a lot about prokaryotic cellular division, and you guys know that prokaryotes divide by binary fission, which essentially means that you have one cell, and that cell is going to copy its DNA, and then you have two identical cells. If I'm being honest, and I always try to be honest, that's not the only way that prokaryotes can reproduce. There is another way that some, and it's not a lot, but some will actually do a process called budding. And with budding, what we do is we have an outgrowth. It's a little out pocket that's very small initially, but then it starts growing. And it grows and it grows and it grows and it grows until it gets about the size of the parent cell, and then it pops off. Can you imagine if that's how we reproduce? Saying, hey buddy, how's it going? And then once they get the same size as you, they just pop off and leave. But that budding process we do see in some microbes, not by any means the majority. The majority is this right here. This is binary fission, where we're going to DNA replicate, which is copy our DNA, and then we're going to divide that cell into two identical cells. Microbes grow at a rate that is considered logarithmic, which means that they grow 10 times the amount every division. So if I'm looking at the fact that there are 100 cells, and I allow those cells to grow for 5 hours, if you look at the scale of the logarithmic scale, and you know that there are 60 minutes in an hour, you can then figure out that in 5 hours, you would have over 1.7 million cells. That's mind-blowing. I think about cleaning, right? We're going to talk about in Chapter 7 some of the things that you do around your house that maybe you don't think about. But I know people who don't vacuum, but maybe once a week. They don't mop, but maybe, I don't know, once every 3 days. They don't dust, ever. You think about the fact that 100 cells, I mean, that's a tiny little area that would hold 100 cells. If you leave 100 cells viable, in 5 hours, you already have 1.7 million microbes. Imagine how many microbes are on your floors if you only vacuum or mop every 3 days. How many 5 hours are in every 3 days? That's insane. People don't realize just how fast these microbes actually reproduce, and it's because they grow logarithmically. They grow in phases. When you first introduce a microbe into the agar, they are in what is called the lag phase. And you can look on this chart right now, and they're not actually growing yet because they've just been inoculated onto the agar. Then they take off. They start growing very, 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 very fast in the log phase. During that log phase, that's when you'd want to hit them with an antibiotic. That's when you're feeling bad, you go to the doctor, they put you on an antibiotic to hopefully stop this growth. Then look up here at the top where you hit the stationary phase. Why does the stationary phase plateau off? Let me ask you this. Are they dying? No. Are they growing? No. So what's happening during the stationary phase that they're not dying or they're not growing? They were growing. And if you look past the stationary phase at the death phase, they start dying out very quickly. During the stationary phase, they've actually utilized all of the nutrients that we have provided for them. They are limited to the agar that we put them in. And so they're going to utilize that, and they're going to be dividing like crazy, like it's a party. But once all the nutrients are gone, they can no longer divide. Now, do they automatically start dying? The answer is no. But once they do start dying, they die just as fast as they reproduce. I'm going to pause here because where we're going to go next is we're going to talk about how we would be able to count colonies and the percentages of microbes that we would have in samples. There's two ways that we can do that, direct measures and indirect measures, that we're going to pick up with next time in this lecture to finish up chapter 6.